We'll go to screen sharing, and we're going to begin looking at the fungi that we were talking about the other day. So let's remind ourselves of that life cycle of the fungi that we were looking at. Oops. So remember, we have a cycle that involves asexual reproduction in this circle over on the left. And then in the right, we have the part of the life cycle that involves sexual reproduction. And notice the part of the life cycle with sexual reproduction is the only part where we have diploid cells, are those weird heterokaryotic cells that are N plus N, where we have more than one nucleus in the same cell, but all the nuclei are haploid. So notice this heterokaryotic cell. Remember, one of the things you've heard me say already several times is, we like to divide things up and put things into nice, clean categories when people look at the living world, or just nature in general, even the physical world. Nature doesn't always cooperate. If you look at these heterokaryotic cells, are they diploid or are they haploid? You could probably make a little bit of an argument for either case. Now, when we talked about the asexual reproduction, we talked about producing spores by mitosis. But before we go on and look at some of the individual groups of fungi, I want to think about two other types of reproduction that occur in fungi that are asexual that diagram doesn't show. So let's look at this diagram first. This is a process called budding. And this is a single-celled organism, a yeast cell. And the pink purplish represents the cytoplasm with all the organelles that are in the cytoplasm. Yeast are eukaryotic cells, so notice that means they have endoplasmic reticulum, Golgi apparatus, mitochondria, so on down the line. The gold, yellow, is the nucleus. So when we look at yeast as well as some other living organisms, some other things that we'll talk about later, can reproduce by budding. And in budding, a new cell pinches off of the original cell. The yeast do have to replicate their DNA, and ultimately we have a small cell that pinches off, and this cell is called a bud. Now, sometimes this bud stays attached to the parent cell that it budded off from. Sometimes it detaches. And if it stays attached, then a bud can form off this bud, and we can end up with a whole chain of cells attached to one another. But since there's no meiosis involved, since there's no horizontal gene transmission, all these cells would have the same DNA. And since they all have the same DNA, that's what makes this a form of asexual reproduction. The real difference between asexual and sexual reproduction is, in asexual reproduction, all the cells that we make from the starting cell have the same DNA as each other, and they have the same DNA as the parent cell that produced them. In sexual reproduction, we end up with cells with different genetic combinations. So yeast reproduce this way, and all the fungi, other than yeast aren't big enough, but all the fungi can reproduce by a process called fragmentation, which means if you take a big piece of fungus, a big mycelium, remember that's the body, and you tear it in half, both halves can regrow into a complete new fungus. You'll kill a few cells, but the problem is, it's not a problem unless you're trying to kill the fungus. The problem is, if you're tearing this up thinking you're killing it, fungus cells are not specialized enough that they can't regrow the whole body. With some organisms, this doesn't work. If you take a person and chop off their leg, you won't have two people when you get done with this. So with a fungus, though, take a small piece, separate it, and as long as it's big enough to survive, it can regrow the whole mycelium again. And the mycelium will be grown by mitosis. So this is a form of asexual reproduction. Because if anytime cells reproduce by mitosis, we get genetically identical cells. And notice when we talk about this, we are ignoring the possibility of mutations that might occur during the replication of DNA when we say that cells in asexual reproduction are genetically identical. There could be a mutation, and we're ignoring that for the purposes of this discussion. So let's look at the various groups of fungi. So one of the groups is the phylum Chytridiomycota, or Chytridiomycetes. And these are mostly aquatic species, which means they live where if they're aquatic? Notice kind of the opposite of aquatic is terrestrial.
Water?
Yeah, so aquatic species live in the water. Sometimes we can specify that more specifically, and we can talk about marine species, which live in salt water, versus freshwater species, which you might find in Lake Michigan. And if we look at this, notice that this group was originally classified with Protista. But if we look at molecular analysis of their metabolism, this suggests that they're fungi. But they're so different from the other fungi, when it was decided that they were actually a type of fungus, not something that was so different it couldn't even be considered a fungus, they were put in their own phylum. So this is aquatic species. They're very primitive. And remember what we said about that? That just means that this is probably the first group that split off from the common ancestor. They still have a lot of similarities with the common ancestor. They don't think of them as being bad because they're primitive. That just means they evolved earlier. So these split off from the common ancestor of all the other fungi. Now, like a lot of cells that we've seen, Chytridiomycota are single-celled organisms. They have a flagellum. And this is really the reason that originally they weren't classified as a fungus, because this is the only group of fungi that have flagella, or cilia for that matter. Fungi don't have flagella or cilia. That was originally one of their defining characteristics. It's kind of outdated now because Chytridiomycota do have a flagellum. There's one particular genus of Chytridiomycota that's very important, and this is Batrachochytridium. This is a fungus, and it infects frogs. And when it infects them, it impairs their ability. It inhibits their ability to absorb water and salt from the environment. And right now, if you go around the world, it's estimated that about 20 or 30% of all the species of frogs are probably endangered. And one reason they're endangered has nothing to do with this fungus. It has to do with the fact that humans are destroying some of their habitats. But this fungus is also killing large numbers of them and making them more susceptible to extinction. So this group does have some important ecological effects. At this point, we haven't really discovered any important medical or industrial applications for these organisms. The Zygomycota or Zygomycetes, and if you're looking these up, our book calls them Zygomycetes, but you might see them described as Zygomycota. Kind of the two names are interchangeable. Another name for this group is the conjugation fungi. So think about conjugation. We've seen conjugation in Paramecium a few days ago. And before that, a few days, we talked about conjugation in bacteria. And in those two types of organisms, conjugation is a form of horizontal gene transfer. Changes the DNA of a cell, but it doesn't create new organisms. In these fungi, it's going to be a little bit different. Conjugation is going to be a form of sexual reproduction. So it is going to change the DNA of the cells that are produced, but also it's going to make new cells. So some of the most common members of this phylum that you might have seen, you probably haven't heard of, are called Mucor and Rhizopus. These are their genus names. Rhizopus is a very common mold. It's typically a kind of a blackish, grayish color. You might have seen it growing on bread or a fruit. Rhizopus is actually a huge agricultural pest because, you know, if you think about bread, bread is made from a grain. So rhizopus grows on a lot of grains, and it sometimes grows on grains that have been harvested and they're being stored in a silo. And there are some species of rhizopus that produce a toxin that's poisonous called aflatoxin. You might have heard about aflatoxin recently. It's been in the news a little bit because there were, I think I heard 2 companies that had to recall pet food that was on the market because it was killing dogs. And the reason it was killing dogs was because there were high levels of aflatoxin in this dog food. So this was produced by rhizopus. Now, even though these are different types of molds, and you might think of a mold as being something very simple, there are actually 3 distinct types of hyphae in the molds in this group. So if we look at this diagram, we have this kind of tannish stuff down at the bottom. This is the substrate. Now, the substrate is just the technical name for whatever the mold is growing on. So this could be a piece of fruit. This could be an animal skin. This could be a piece of bread. It could be a branch on a tree. And what we see is that, remember, the hyphae are the different strands that make up the body. There are some hyphae that run along the surface of the piece of bread or the fruit or the branch of the tree. These are called stolons. Now, if you were looking at a mold and this mold had grown enough so that it was visible, because if there's just a few cells, it's really so small you can't see it. But if you were looking at a piece of mold on a fruit in your kitchen, what you're seeing is mostly the stolons. This is the part that's visible. There are some parts that go underneath the substrate that kind of push through the substrate. These are called rhizoids. And you could think of rhizoids as being like the roots of a plant. They really have 2 functions. They anchor this mold into its substrate, whatever it is. And remember, fungi are heterotrophic by absorption. So these rhizoids release digestive enzymes into the fruit that this mold is growing on, or the piece of bread, or the tree branch. And they dissolve some of those cells, which they then absorb for nutrition for the fungus. Now, under certain circumstances, they then make a type of hypha that's called a sporangiophore. These sporangiophores have different names in different groups, but this is a stalk, a hypha that sticks up off the surface. So the stolons run along the surface, this sticks up. Now keep in mind, when I say this sticks up off the surface, we're still talking about a fraction of an inch, maybe an eighth of an inch at the most. So this is not something that's real noticeable. And notice this sporangiophore, the P-H-O-R-E ending. When we see that P-H-O-R-E ending in general, that's going to be a hypha, and it's going to be a hypha that has spores at the end of it. Now, this sporangiophore ends in something called a columella. And this columella has a sporangium that grows off of it, and the sporangium divides and produces spores. And the sporangium produces the spores by mitosis. So, you know, if we go back to the life cycle we were looking at before, we're really looking at this structure right here, the spore producing structure that's N. So we're in this part of the life cycle over here on the left. We're in the asexual part of the life cycle. Now, when we look at these, notice that these stolons and these rhizoids and these sporangiophores are sometimes considered to be organs. So in that sense, this is a fungus that does have organs. Now, if we look at Rhizopus, here we can see some Rhizopus magnified under a microscope. So all these thin little green strands are hyphae. Depending on how they collected this, they could be stolons, they could be rhizoids. It's not really easy to tell from this micrograph. But every once in a while, we can see a stolon. Let's follow this stolon here. Notice it goes here and then it bends and it leads into this structure. So this stolon is a sporangiophore. And this lighter area in this circle, this is that columella that we were just talking about a moment ago. And then this is the sporangium with the spores, this darker area. Now, a lot of times when you see the sporangium under a microscope, it'll be kind of torn up because it's actually pretty big in a three-dimensional sense. It's big enough that it's hard to put on a microscope slide. The other reason it might be torn up is because eventually, as it produces spores, this sporangium bursts and releases the spores. So the reason that this is typically an elevated structure is these spores are going to drift on the breeze to their next substrate. And they're going to germinate on that next substrate when they get there, assuming it's something they can live on. They're going to germinate, they're going to start dividing by mitosis, and they're going to make a new mycelium. So if we get them up in the air a little bit, these spores have a better chance of dispersing to a new substrate. You can imagine if this sporangiophore was very short and our columella and our spores were down here, they fall right on top of the current mycelium. It wouldn't be very good. So these are typically produced so that this is why if you have, you know, some mold growing on a piece of bread, you might throw that piece of bread out and a day or two later you find mold growing on another piece of bread. Why? Because the sporangiophores had already released some spores. Plus, these are pretty delicate structures. So it's pretty hard to pull one bread out of the sack of bread without breaking some of these open. Now, one of the things to notice, and this is true about many fungi, if we would look at all the stolons and all the sporangiophores and all the rhizoids, and we could collect them all and we could weigh them accurately, it turns out that most of the fungus is actually in the rhizoids. Most of the fungus is underneath the surface of the substrate. So when we think about a fungus, one of the things we want to keep in mind is a fungus in some senses is similar to an iceberg. And it's similar to an iceberg in the sense that, you know, for an iceberg, we say that 90% of the iceberg is underwater. Most of the fungus is under the surface of the substrate. So if you have athlete's foot, if it gets pretty bad, you might be able to see some actual fungus growing on your toes. What you can't see is most of the fungus that causes athlete's foot is actually inside your foot. So the fungus cells are growing in between your cells. And this is why there are some products to get rid of athlete's foot now that advertise that they work on the inside, because on the outside, you can kind of scrape athlete's foot off. You can use a nail file or something and you can scrape it off. That doesn't get rid of athlete's foot. It'll come back the next time conditions are good, which for fungi usually means warm and moist. So when does athlete's foot come back typically? After a bath or a shower, where there's been some moisture in the environment. So we want to keep in mind that most of the fungus can't be seen because it's underneath the surface of the substrate. So here's our rhizopus. This type of rhizopus, where it has the asexual spores, is sometimes called Rhizopus vegetative. When we talk about things that are vegetative, we're talking about things that are reproducing asexually. So for instance, grass that might be growing in your front yard can reproduce with flowers, but usually it just spreads through underground runners. That's a type of vegetative reproduction. So now this is magnified a little bit more, but this is what Rhizopus looks like when it reproduces sexually. So let's focus on this structure down here. So notice we have this dark structure right here. This is the zygote. And the zygote is produced when we have two mating strands. So here's a cell from one mating strand on the left of this. Here's a cell from another mating strand on the right. So these two mating strands grow together and they fuse to make the zygote. Now, remember what we've said about the life cycle of fungi. The life cycle starts out, the zygote starts out as a heterokaryotic cell. So it starts out N plus N because we get one nucleus that's haploid from the cell on the right. We get one nucleus that's haploid from the cell on the left. Then these two nuclei fuse. When the cytoplasm comes together, that's plasmogamy. So think of plasma at the beginning of plasmogamy, cytoplasm. When the nuclei fuse, that's karyogamy. And then the zygote, when these nuclei fuse, is now very briefly 2N. It's diploid. Now, once it's diploid, this zygote divides by meiosis and makes 4 spores, which are called zygospores. So lots and lots of different types of spores in fungi. And in general, they're named for the structure that produces them. Now, if we would be looking under a microscope, we would see some zygotes like this zygote here that doesn't look like it has any hyphae attached to it. And that's just because, again, this zygote is a relatively large structure three-dimensionally to be putting on a microscope slide. So when we put it on there, a lot of times we tear the hyphae off. Or you might see a zygote like this one here, where I can see one of the hypha cells that's attached to it. I can't see one on the other side. One of the things we have to be cautious about when we look at things under a microscope is what they are really appearing to be might not be what they truly are. Some of these are what we call artifacts. Artifacts, when we're talking about a microscope slide, are things that are produced by the way we make the slide. They're not really there. You might remember that Camillo Golgi, the Italian scientist who discovered the Golgi apparatus, when he did his first work, everybody thought he was just creating an artifact when he looked at cells because nobody else could stain the Golgi apparatus correctly so they could see it in cells. So when he was talking about this, everybody just thought his microscope technique wasn't very good. So that's something we always have to be cautious about. So notice that this zygote has a very thick cell wall. That's one of the reasons that it's so dark. When we look at things under a microscope in general, things that are dark absorb a lot of the light. So we don't see much light through them. Things that are more lightly colored don't absorb as much of the light. The cell walls are so thick here that this zygote can survive for several months. So a lot of the organisms in this group, the Zygomycota or Zygomycetes, make spores towards the, if we're talking about in this part of the world, they make spores in the fall. and then the zygote overwinters. And then the next winter, the zygospores are released and the new mold starts growing. Now, a group of fungi that used to be classified in the zygomycetes are the glomerulomycetes. Or again, they're sometimes called glomerulomycota. Mycetes and mycota both mean fungus. A mycologist is somebody who studies fungus. They form something called arbuscular mycorrhizae. Arbuscular, think of Arbor Day. Arbuscular means they're tree-like. Mycorrhizae, well, remember we just said myco refers to fungus. Rhizae, think of those rhizoids we just saw. Rhiza means root. So these are root fungus. And although we don't normally eat these are crucial for human life. Without the fungi that are found in this group, plants would really have a very limited ability to grow on the land. Plants would mostly be restricted to aquatic environments. And these are found in about 80% of all land plants. And they do two things. Remember, fungi get their food by absorption. So what do they do for a plant? They absorb minerals from the soil and they fix nitrogen. And then they make these materials available to the plant. So even though when we talk about plants, we say that the root absorbs minerals from the soil, a lot of that absorption is really done by the fungi that live in the roots. Now, in this case, these fungi make branched hyphae called arbuscules. And so if we look at a plant, remember what we said about fungi before when they're growing on a substrate. Most of the fungus body is actually below the surface of the substrate. And when we look here, we have the fungus on the outside, and then the fungus actually grows in between cracks in the cells. And this would be the root of a plant here. And then it makes an arbuscule. We aren't going to look at all the details of the structure of an arbuscule, but this arbuscule is actually inside a plant cell. Now, what does the fungus get out of providing all these minerals and all this nitrogen to the plant? The plant gives the fungus as much as 20% of the organic molecules that it makes during photosynthesis. It's usually a lot less than that. But this means that the fungus basically, in exchange for providing these raw products to the plant, gets back in return some carbohydrates, mostly carbohydrates, but a few proteins and lipids as well. So when we look at this group compared to other fungi, all of the fungi in this group pretty much live the same way. They all form these arbuscules with plant roots. In most of the other fungi, there are several different groups that survive in slightly different ways, but not so with this group. And as they say, this was once a subgroup of the zygomycetes, that first group that we talked about with the rhizopus. Another phylum of the fungi is the basidiomycetes, or Basidiomycota. And they're called the club fungi because they make a structure called a basidium. And I don't know, if you squint real hard and use your imagination, somebody thought a basidium looked like a club. We'll see a photograph through the microscope in a little bit. I'm not sure I see that, but to each their own. This includes a lot of common species. This includes some things called puffballs. It includes mushrooms, so all the mushrooms that we eat. It includes toadstools. And we all know how toadstools got their name, right? They're kind of a specialized group, very similar to mushrooms, but old legends say that, when all the people leave the forest, the toads come out and they hold council and they sit on the mushrooms. Those are their seats. There are some things called bracket fungi and rusts, and smuts. And all of these things, bracket fungi and rusts and smuts, these all grow on plants. Some of them mostly grow on plants after they're dead. Some of them are parasitic and grow on plants while the plants are still alive. And it also includes some ectomycorrhizae. Now remember, we talked about the arbuscular mycorrhizae before. Ecto means outside. So this is a fungus that lives in association with a root, but it lives outside the root, which, as we'll see as we've learned more about these fungi, turns out to be not completely true. Now, since the rusts and the smuts and the bracket fungi are often parasitic, this means that, like most parasites, they have very complex life cycles. So they're fairly economically important because a lot of them actually attack plants that are crucial in agriculture. So let's look at a diagram of the life cycle of in this case, it's a rust. So this is a type of rust called wheat rust, sometimes also called stem rust or black rust. Usually lives on the stem of the wheat plant, or it's black in color. But if we look at Puccinia lives on two different hosts. One of the hosts it lives on is something called a barberry plant. And barberry plants are common ornamental plants. A lot of people have them around their houses. And it's basically a type of bush. There are other barberry plants that are not very bushy. So we're going to start out with something called Basidiospores. Notice this is a group, Basidiomycetes or Basidiomycota. So Basidiospores land on a leaf of this barberry plant and they germinate. Now, the Basidiospores are haploid, so they're one N. Now, there are two versions of these, and the Basidiospores fuse in a process called plasmogamy again. So remember what we said, plasmogamy is the fusion of the cytoplasm. And when these spores fuse, this makes a structure called an acium. So the acium is this kind of U-shaped structure. We'll look at one in a few moments. And the acium divides and makes aciospores. Because Puccinia is a parasite and it goes through several different parts of the life cycle, think about when we talked about that Plasmodium protista the other day that causes malaria. Remember, we had the sporozoites and we had merozoites and we had the oocyst and the ookinete. We had all these different stages. We're going to have tons of different spores here. So we make these aciospores. Now, the aciospores get carried on the wind to a wheat plant. And while they're on the wheat plant, this is showing a little stalk, and it's probably hard for you to see unless you've copied the notes already. They have little red and black splotches on them. So that's why this is wheat rust. Now, when these aciospores land, they form a structure called a uridinium. And the uridinium is all this kind of orangish that's flat down here. And the uridinium makes uridiniaspores. And the Uridinia spores are very distinct because they're kind of pointed. And these Uridinia spores can be blown by the wind, and they can either reinfect a different part of the same stalk of wheat, or they can blow to another wheat plant in the same field. And when I say early in the season, you might wonder, how can a fungus tell that it's early in the season? Well, the way that the fungus can tell is temperature changes create certain physiological changes in the fungus. So as long as the temperature is relatively warm and nice, these Uridinia keep producing Uridinia spores, and they reinfect the wheat plant. But late in the season, so think about if we're here in Illinois, temperatures start to decrease. These decreases in temperature cause certain chemical changes in the Uridinium. And the Uridinium starts producing teliospores, and the teliospores are 2N. So these teliospores fuse together because the way that they fused was through karyogamy. And remember, we had cells over here that were N plus N. So when they fuse, we now have 2N cells. They divide by mitosis, and they make a basidium. So here's a diagram of a basidium here, or up here. Here's another one. And the basidium have 4 spores attached because, pardon me, I think I said the basidium divides by mitosis. I should have said meiosis. So when we have a cell that divides by meiosis, we get 4 sperms, 4 spores, and these are the basidiospores. Now, because they're made by meiosis, they're not all identical. And these basidiospores then go back to the barberry leaf where they land and the cycle starts again. Now, these telia, again, notice it shows them with very thick brown lines around them. These telia, again, have very thick cell walls. So this stage can potentially survive over the winter and then infect the wheat crop when it comes up the next spring. So this organism has a way of kind of avoiding the climate changes or the weather changes that potentially could kill it. But notice we have basidiospores, we have aciospores, we have Uridinia spores, and then we have teliospores. So we have 4 different types of spores in this life cycle. I guess I should point out these Uridinia spores are sometimes just called Uridia spores. So let's look at some of these in real life. So here is a misspelled, I had to add an A at the beginning. This greenish structure here, this is this acium that we can see right here. And it has lots and lots of spores in it. Notice that once this acium breaks open, then we see something that has a very typical kind of U shape to it. And when it breaks open, now these aciospores spill out and they can be carried by the wind to a wheat plant. Notice that when we think about this, there are a lot of spores in this acium, and we can only see part of it, but there's another acium right next to this. Why so many spores? Well, keep in mind, if you're a parasite, if you're this Puccinia and you're living on a barberry leaf, what are the odds that this spore that's traveling, it's getting blown by the wind, so it doesn't have any maneuverability of its own? What are the odds it's going to land on a wheat plant? So when we have something that reproduces through a process that's not very directed, we'll see this when we look at a lot of plants that are wind pollinated. If the wind is going to blow your pollen, or in this case, your spores, to the next part of your life cycle, you better make a lot of them to make sure they get to the right place. So we have a lot of acaeospores. Now, they land on the wheat leaf and they form a uridinium, which is this structure right along here, just kind of the base. And they grow underneath the outer layer of cells on the plant. So if we look at if we look at this flap here, and if we look at this flap here, this is a slice through a stalk of wheat. What we see is A is actually the epidermis of the plant, because as the uridinium starts to grow, it gets so big, it basically causes this epidermis to pop. So this epidermis peels off the surface of the plant. And then it starts making the Uridinea spores here. Now the Uridinea spores, and I'm going to see if I can increase our magnification quite a bit for just a second so we can look at these. These spores, if we look at them, are kind of nice oval spores. And the reason I wanted to increase our magnification so much is we're going to compare that to the teleospores that we see in a moment. Now, these are the only type of spores that can infect the same type of plant that they're on. Those acaeospores that we talked about a moment ago are produced on a barberry plant, but they can only infect a wheat plant. These are produced on a wheat plant and they can infect a wheat plant. So these are the spores that I mentioned above that can spread to another part of the same plant or to another plant in the same field. And then finally, we make these teleospores. Now with the teleospores, remember we said earlier, we have two cells that are both N, and they are going to fuse and then divide by meiosis. On a lot of these, you can see the two cell structure. Usually one cell is slightly lighter, one cell is slightly darker. The other thing you can also notice on a lot of the telea that are going to make those spores is they have kind of a pointed end. They're not nice and oval. So wheat rust is a very important parasite. If we look worldwide, depending on where you are in the world, fungi can destroy anywhere from about 5 to as much as 20% of the grain products that are grown. So considering that in many parts of the world, people don't get enough food, this is a huge problem. Now we mentioned that some of the Basidiomycota form ectomycorrhizae, mycorrhizae that are on the outside. So basically the ectomycorrhizae do the same thing as the arbuscular mycorrhizae. They both absorb materials and make those materials available to the plant. Well, notice if we start out looking on the left side, we can see some plant cells. The cells are these white cubes, squares, and the cell wall is the kind of the beige or the green in between them. So if we look on the right, here's one of those arbuscular mycorrhizae. So they have a structure called a spore out here. And if we look at the hyphae, notice that the hyphae go right through the middle of these white squares and rectangles. They go through the plant cells. And then they form this arbusculae, which has these little branches. So that's why, remember I said arbo means tree. That's why it's an arbusculae. And these trees are inside root cells. If we look on the left side, it shows ectomycorrhizae. And the blue is the fungus here. Notice most of the blue is on the outside of the plant. And there is some part that goes into the plant, but notice the part that goes into the plant grows between the cell walls of adjacent cells. If we look inside all these white rectangles and these white cubes, the blue is not inside them, it's between them. So in the ectomycorrhizae, the fungus doesn't actually penetrate the plant cells. Now, the other group of Basidiomycota that we have is most of the things that we think of as resembling mushrooms. A lot of mushrooms are edible, but notice some of them are poisonous. Some of the ones that are poisonous because they produce hallucinogenic chemicals. And, you know, if you ingest a small amount, you might go on a trip. If you ingest a large amount, it might kill you. When we look at a mushroom, think back to the molds we were discussing earlier. The mushroom is basically the reproductive structure of this fungus. Most of the fungus's body is underground. This is one reason why if you have mushrooms that grow in your backyard after a heavy rain, you could pick the mushrooms and you might think that they're not going to come back, but most of the body of the mushroom is actually underground. So that's not true. So when we look at mushrooms, since most of the body is underground, they're often growing on the roots of dead trees. And since roots are roughly round, when you look at how the mushrooms come out, they come out in a fairly round pattern. And this pattern is called a fairy ring. And again, like toadstools, because, you know, when all of us big folk get out of the forest and the fairies meet and hold their conferences, where do they go? They all sit on a mushroom in the fairy ring. The hyphae are septate, they're divided by cell walls, but a lot of times the cross cell walls are perforated. They have holes in them. And these holes basically allow materials to pass from one cell to another. So let's look at an example of a life cycle of one of these organisms, but I think we're about out of time today. So we'll wait and we'll pick up with mushrooms on Monday. So later today, I should have your test scores posted and hopefully I'll have some papers read too. And I can send you my comments on your papers. One thing to keep in mind that I have noticed from some people, if you send me an email, make sure that, you know, some form of your name is on that email. Some people are sending me emails and, you know, your email address doesn't necessarily look very similar to your name. So you want to make sure you put your name on that. The same thing applies when you turn in assignments. You want to make sure that, you know, there's a spot up at the top for your name. So make sure you type in your name, not just the answers to the questions. That would be very helpful. Are there any questions before we leave for the weekend? No? Well, I will stop recording.